Hello students, last time we have studied the quantum theory of Raman effect, and we have also seen some important characteristics of Raman lines observed in the Raman spectra. Now in this next video I am going to explain the experimental setup of Raman effect. So the kind of instruments or the kind of setup required to study or to observe the Raman spectra of the given sample. So what are the basic devices or instruments needed to observe such spectra that we are going to study here. And finally, we are going to discuss some of the important applications of Raman spectra or Raman spectroscopy. Okay, so the experimental setup of Raman effect. Now, what is the basic principle in Raman effect is that the sample for which you want to study the Raman spectra must be illuminated with high monochromatic source of light. So, initially the sample must be illuminated with the high monochromatic source of light, and the scattered radiation, whatever the interaction of these radiation with the sample, they should be recorded. Okay, so the scattered radiations, they are recorded by arranging spectrograph in transverse direction. So the direction of incidence on the sample and the direction in which they are collected, that means the scattered radiation are collected, they are mutually perpendicular to each other. Now, the original arrangement that which was used by Dr. C.V. Raman, it was not much efficient because the kind of technology available at that time and the required sources for the different applications, they were very limited, and therefore the time which was required to get a good record, so very long exposure of the order of about 100 hours was needed to get a good spectra or good record of the given sample. So later on, many scientists have made improvements in this setup required for Raman spectra, and the first very fundamental improvements which were developed by the scientist Wood, and the kind of Raman spectroscopy that nowadays we are using is very advanced. So to obtain a good Raman spectra, what is required is that the incident beam should be very strong. Why it should be strong? Because Raman scattering is very weak. Okay, so the Raman scattering is very weak, and if you want to get sufficient shift, that means the intensity of the Raman line sufficiently, then the incident radiation should be very strong. And one more requirement, that the incident radiation must be highly monochromatic, because Raman lines are observed on both sides of the central line, which is a Rayleigh line, and the spectral difference, if you see, between the Rayleigh line and the what we call Stokes line and anti-Stokes line, it is very small. So the spectral difference is very small, and therefore we need a highly monochromatic source of radiation. If it is not highly monochromatic, then what will happen? The small shift in the frequency or wavelength, that cannot be observed clearly in the Raman spectra. So this is the basic requirement, two very important requirement before going to the actual study of experimental setup. One must understand these two requirements. Okay, now let us see the main components involved in the experimental setup of Raman effect. So I have shown here the diagram, and some letters are used to represent each of the particular block in this diagram. So this is simply a block diagram of experimental setup to observe the Raman effect. Now you can see this is the first block, that is a laser source. So laser radiations, you know that these are highly monochromatic and their intensity is very high. They are well focused. Uh, Therefore, the beam can be a suitable source of radiation in order to observe the Raman effect. So, this is a laser source. Then filter. So, even if the radiations have some components, additional component that can be filtered out other than the required wavelength, and in some cases the filters are also used, cooling filters are used uh, in order to avoid heating of the sample. Then this is the focusing lens. So the incident radiation, they are properly focused on the sample. Okay, L1 is the focusing length and C is the sample cell. You can see here C is the sample cell. Later on we are going to discuss more about each of these components and what type of sample cell is used and all that. So we shall discuss later on. Then this is the mirror. So this is the optical system consisting of lens L1, L2 and this mirror. So in order to lens L1 is used to focus the incident light on the sample cell, and the mirror also, whatever the transmitted light, it will be reflected back again towards the sample. So maximum radiations will be utilized in order to illuminate the sample. Okay, then the scattered light, it is collected in a transverse direction. You can see this is the incident direction and this is the transverse direction. So the scattered light is collected in the transverse direction and the collecting lens properly focus these scattered radiation on the spectrophotometer. The SP represent the spectrophotometer. Now this spectrophotometer, you know that it is a kind of photo detector. Okay, so the incident photons, they are actually analyzed in terms of their intensity and frequency. The incident photons, they are count as a function of their frequency, and this data is given to the recorder. So this is the initial the electronic recorder, and the incident the signal received from the spectrophotometer, it is converted into electrical signal. There are some sensors just like a photo detectors, and these electrical signals are amplified and these are recorded by the electronic recorder as a plot of intensity of the radiation against their frequency. So this is the exact nature of the experimental setup of the Raman effect. So these kind of devices are used. So in the modern experimental setups, some component may not be present or some may be additional component like computerized spectrographs are available now, and the software and the hardware to interface the actual devices. These are some additional components involved in a very latest form of Raman spectrographs or spectrophotometers. Okay, so the experimental setup of Raman spectrometer, that mainly consists of a laser source, okay, which is highly monochromatic. The optical system, as I have mentioned, the filter F to filter out additional components present other than the required wavelength, the lens L1 focusing lens on the sample, and mirror M to focus the light on the sample, and there are some collecting lenses, for example L2 to feed the scatter radiation to the spectrophotometer. Now the spectrophotometer counts the photons as a function of frequencies. Then this analysis is delivered to the as a plot of scatter intensity versus frequency by the recorder R. Okay, now this is the typical Raman spectrograph. Okay, you can see here a plot produced by this recorder. It look like this. Okay, this is the intensity against the frequency here. Now if you see these three lines, as earlier we have discussed in the Raman spectra, the incident radiation, whatever the frequency, that will appear as a central line, the same frequency, and it is called as a relic scattered line. Okay, and it is of the same frequency, and this is the excitation frequency. Let us call it as an excitation frequency. Then anti-Stokes line and Stokes line. Anti-Stokes line, which are having slightly greater frequency than the relic line, and Stokes frequency, they are having slightly less frequency than the relic line. So anti-Stokes line, you can see the frequency the excitation frequency plus new vibration. Actually, here new vibration is the vibrational frequency. Okay, the interaction involved between the incident radiation and the vibrational motion of the molecule in the sample. So that causes this uh, addition in the frequency, new excitation plus new vibration. And there may be a chance that this is called as a vibrational anti-Stoke Raman line. There may be chance that the rotational energies, they are having interaction with this particular, there is an actual transition of the energy of the molecule from the initial level to the this additional new excitation plus new vibration. So similarly, it can be possible for rotational levels also. That means new excitation plus new rotational. So in that case, the Raman lines will be called as the rotational Raman lines. Okay, either rotational anti-Stoke line or rotational Stokes line. In that case, you have to specify this as new excitation minus new rotation, or new excitation minus, sorry, plus new rotation. Either rotational energy or vibrational energy may be involved in a particular interaction. Okay, so this is the kind of typical Raman spectra one can observe. Now let us discuss about some important components of the Raman spectrometer. What kind of laser sources they are used for the Raman spectrometers? That first of all we shall see some of the examples. So the commonly used laser sources are helium neon having wavelength 633 nanometer, then argon ion laser having wavelength 488 nanometer, then HgCd gas laser having a wavelength of 325 nanometer, ruby gas laser having wavelength of 694 nanometer, then ND-YAG solid state laser having wavelength in the range of 1.06 micron. Then tunable dye lasers, they are also used, which are in visible range from 0.43 to 0.65 micrometer, and the UV lasers are also used in some of the Raman spectroscopy applications. For example, 184 to 260 nanometer. Now the power of these particular laser sources, depending on what type of laser source you are used, it varies from 50 milliwatt to almost 2 watt. So that depends what type of source you are used. Now, what are the advantages of using laser source? Why we are using laser source? Earlier we have seen that since laser radiations are highly monochromatic, then they are having high intensity. In addition to this, there are some more advantages of using laser source. First and very important thing, there is no need of cooling the sample, because the radiations, their intensity and the focus can be properly controlled. So no need of cooling here, and the filter is also not required because they are already highly monochromatic. No additional components are present in the source. Then the amount of analyte required if you use a laser source, that is very small, because you know the focused they are very convergent. The source, laser source, is very highly focused source, so very small amount of analyte is required. Sample required is very small in amount, therefore you can save your valuable samples. Then second order Raman spectra can be also recorded. This is very important in some applications. This second order Raman spectra can give you valuable information, and it can be recorded if you have highly monochromatic laser source. Then in some cases the incident radiation may have interaction with the vibrational mode of the molecule because of vibration. So you know that because of the relative motion between the source and the object, you will get the modification in the frequency, and that is called as a Doppler effect. Okay, apparent frequency. So therefore the actual source radiation will be appear broad. So that broadening due to Doppler effect can be minimized because your frequency source is very sharp. Okay, having a very small bandwidth, you can say. Then the polarization of laser is well defined and can be controlled within 0.1%. You know that the incident radiation must be having specific polarization. Okay, so the most of the laser radiations, they are plane polarized and they are well defined in a particular plane, and they can be controlled even within the accuracy of 0.1%. So there is no much deviation in the polarization of the source, and therefore the actual interaction involved will not be having any error because of the variation in the polarization of the source. Okay, so the width of the laser line is very small, which is of the order of 0.005 centimeter inverse. So spectral width, you can say in terms of wavelength, or you can say in terms of a wave number, exactly. So you can obtain a very precise information if you have such a very smaller width in terms of the wave number of the given source of radiation. Then there are some basic requirements or basic important facts about the kind of sample cell required in Raman spectrometer. So what are these requirements, or what are these specific characteristics of the sample cell that we shall see here. Okay, so first of all, the typical material that is required for the sample cell is, it is made up of non-fluorescent fused quartz material, and it has well polished bottom, because from the bottom side the radiations are incident onto the sample. So the sample is kept in this non-fluorescent fused quartz tube. Mostly it is a rectangular long tube, and the top of the cell is sealed with a transparent cap to avoid dust and designed to permit deoxygenation of the sample. Then the cell is completely filled, because if you keep it empty, the radiation may reflect from the surface of the liquid inside the sample cell. So in order to avoid the reflections from the sample surface, the cell should be completely filled. Then the spectra of liquid sample is recorded by focusing the beam vertically through the bottom window into the sample, and scatter radiations are collected in a perpendicular direction of the cell. So we have already seen in the actual experimental setup diagram. Now if you have a solid sample, because the Raman spectra can be taken for any state of matter, solid, liquid, gas. So for solid samples, capillary is used to fill the sample. So you know the capillary is having very small diameter, so you need a very small amount of analyte, and the sample is filled, it is sealed at one end, and the laser beam is directed along the length of the capillary, and in the transverse direction the scatter radiations are collected. In some applications, the samples may get degraded because of the laser radiation. They are sensitive to laser wavelength. So in some cases, in such cases, you need to take care. So what type of care is taken, or what type of sample cell is used? So in these particular cases where the sample gets degraded when it is exposed to laser radiation, so in such cases rotating cell technique is used. So that will avoid continuous irradiation towards the laser source of the sample. Some of the samples which are sensitive, like CCl3 or COCl. So in such samples, in order to obtain Raman spectra, we are using a rotating cell technique. So that it is not continuously exposed. The sample is not continuously exposed to the radiation if the cell itself is kept rotating. Then suppose you have biological samples. You know that biological samples are also very delicate, they are very sensitive. So local heating because of incident radiation may degrade the molecule. Therefore, the kind of spectra that is recorded is different here. Resonance Raman scattering of laser radiation from vibrational mode of biological molecules are specifically recorded if the sample is biological. Okay, so this is all about the requirements of the kind of source required and the type of sample cell that is required. Now, finally, let us go to the last topic in this particular chapter, that is applications of Raman effect. We shall discuss here some of the important application. Now, first application, that these Raman spectra helps us to predict the molecular structure, to study the molecular structure, what type of structure the molecule possesses, that can be predicted by using the Raman spectra. So it is very useful to study the structure of molecule. The important information that we get from the Raman lines, their intensities and the state of polarization, we can use to predict the structure of the molecule. So for molecules, for example, having covalent bond. So what is actually bond? Bond is actually nothing but the force or the kind of interaction between the two atoms which hold them together as a molecule. So why it is important here? The molecular structure, it is actually the kind of interaction or forces involved between the different atoms which hold them together, and that actually forms the typical structure of the molecule where the potential energy is minimum, okay, the stable structure. And that is possible here. How Raman spectra helps to study the molecular structure, because you know that basically the every molecule has its own natural frequency of vibration. Okay, so we have seen that nu zero is equal to 1 upon 2 pi under root k upon m. So what is k? K is the force constant. So force constants are related with the bonds or bond strengths, and the m is the mass actually. For diatomic molecule, we have seen it is the reduced mass. Now this particular interaction, the frequency of vibration, that is the natural frequency of molecule, and the incident radiation. So the kind of Raman shift that we get, it is function of these strengths also, or these bond also, or these functions also, these forces also. So therefore here the strength of the bond or the forces involved in the bond, these are also very important, and that will help us, Raman spectra help us to understand the nature of these bonds or the kind of forces exist in a molecule, or indirectly you can say we can study the molecular structure. So for example, here if you see the covalent bond, the polarizability in case of covalent bond changes due to nuclear oscillations, because you know that in covalent bond the electron is shared by the two atoms. So because of the nuclear oscillations, the polarizability of the molecule changes, or there are changes in the molecular polarizability, and this gives rise to intense Raman line. So interaction is very strong, therefore we are getting the intense Raman line. Whereas if you see the different molecule, for example having electrovalent bond, then the binding electron change over from one nucleus to another. So you know that in electrovalent bond, the actual electron is transferred to the other atom for the formation of molecule. In such case, polarizability is not affected by the nuclear oscillations, and therefore the Raman lines doesn't appear. So if for a molecule Raman line doesn't appear, you can directly say there is a electrovalent bond, and if it is very intense, you can say it is a covalent bond. So like this, the nature of the bond can also be studied. What just I want to point out here is, while studying the Raman spectra, you can find out you can predict the nature of the bond or the forces exist in the molecule. If suppose another example I can give you, we have a triatomic molecule just like water, H2O. Okay, so two hydrogen atom and one oxygen atom. So Raman spectra tells us whether these molecules are symmetric, asymmetric, linear or non-linear. For example, here the water molecule is symmetric, but it is non-linear. Okay, and the angle is 120 degree. You know that. So that can be predicted from the actual Raman spectra. So from the number and intensity of observed lines in the Raman spectra, we can have important conclusion regarding the molecular structure. Then a second important application is the nature of the liquid state. Okay, so what is actually nature of the liquid state? So every a liquid has its own molecular structure, and these, because of this molecular structure, number of physical and chemical properties are changing. Okay, so the physical property possessed by the liquid or the chemical properties possessed by the liquid, they depends upon the molecular structure. For example, the physical properties, or let us say mechanical properties, like the capacity to transfer hydrostatic pressure, okay, the elastic waves and all that, or the viscous resistance, that means viscosity, and the tension exhibited by the free surface tension. So all these, they depend upon the molecular structure. So when you predict about the molecular structure by studying the the Raman spectra, that means you are predicting the nature of the liquid state. Okay, so this or you can also predict the physical chemical properties of the liquids in terms of their molecular structure. And this is the major problem in physics, and one can study this with the help of the Raman spectra. So observation of Raman spectra of liquids may lead to important conclusion regarding the nature of the liquid state. Then in crystal physics or in crystallography, you know that there are enormous techniques, just like X-ray diffraction is very important tool to study the the crystal structures. But there are some informations which cannot be even revealed from the X-ray diffraction patterns about the crystals. So such information can be retrieved by studying the Raman spectra. So what are such information? For example, the strength and nature of forces which hold the crystal together, that can be understood by the kind of Raman spectra and not by X-ray diffraction spectra. Then existence of mosaic structure and thermal agitation in crystals can also be studied by the information that we retrieve from the Raman spectra. Then there are some applications in nuclear physics also. The study of spin, statistics, and isotopic constitution of the nucleus can be studied by the Raman spectra. Then study of rotational Raman lines. See here, rotational Raman lines. So as I have already mentioned that the Raman lines can be rotational Raman lines or vibrational Raman lines, depending upon the kind of interaction. So the Raman shift can be by an amount of vibrational energy level difference or by an amount of rotational energy level difference, because of the typical interaction of the molecular rotations or molecular vibrations with the incident radiation. Okay, so the study of rotational Raman lines of homonuclear diatomic molecules such as H2D2 means deuterium, O2 and N2. So such a homonuclear diatomic molecule, the rotational Raman lines can be studied. And in nuclear physics, you know that deuterium and their properties, their applications, they are very important in nuclear physics, and that can be studied by Raman lines. Then in chemistry, in a broad way, you can define the applications. For example, in physical chemistry, the state change from crystalline to amorphous, that can be studied by the Raman spectra. Then some of the phenomena like electrolytic dissociation, hydrolysis can be studied by the Raman spectra. Then in organic chemistry, we can use this Raman spectra to determine the presence or absence of linkages in a molecule, and one can also use this to identify some impurities of certain types. And finally, the biological applications, Raman spectra is also very important in some of the biological applications. So for example here, it can provide us information on peptide backbone disulfide breach and environment of some side chains like tyrosine and histidine. Then it is also useful for study of structural and conformational changes and denaturation of macromolecules like protein and carbohydrates. You know that protein and carbohydrates, they are very long chain molecules, and for this molecule you can use Raman spectra for the study of structural and conformational changes. Then a study of structural and conformational aspects of lipids, then biological membranes and lipid protein complexes, they are also possible by the way of studying of the Raman spectra. So in this way, we have completed this particular last chapter also, where we have studied what is Raman effect. Then we have understood the Raman effect on the basis of classical theory. Then we also discuss the quantum theory of Raman effect. We have studied some important characteristics of Raman lines, and finally today we have studied the experimental setup of Raman effect and the actual applications of Raman effect also we have studied. So we have completed the entire syllabus of this paper 4th at T.Y.B.Sc semester 3rd.